And then I'm basically trying to like tease out what's right here without bringing other stuff. Hang on. So I'm going to hold it right here for a second. The brain looks like two Mickey Mouse ears and a tail. And so in this glob, it's right here.
Wow.
So it's really hard unless you know what you're looking for.
Or
But
yeah.
we got textbooks and all of that. So I know that what I'm looking for is one lobe. The second lobe is somewhere under here. Next to this are wing discs. This might be the other lobe, and that is the ventral nerve cord. So this altogether makes up the fly's central nervous system, and that's why we use it as a model. But yeah, and then so what I just did was gross dissection. You would then go back through and like get these things off and just have a cleaner brain.
Right.
For the day, these are going like for say immunofluorescence experiment or maybe like sequencing. And based on what you're doing, it may or may not have to be as clean.
Why are you looking at fruit fly brains? Like, what are you actually studying in terms of brain and development over time in the brain?
Yeah, great question. And honestly, the most important question. I guess we should have started there. But the big picture question for me, my project, and kind of where I think a new direction of our lab is going is how does the brain know how big to grow? And so I'm studying, of course, we know the central dogma that is DNA, RNA, protein, but I'm looking more at the post transcriptional regulation level, and I'm looking at an RNA binding protein, and more so just asking the question, say in neurodevelopmental disorders where the brain size is messed up, you have macrocephaly, which is when a human baby is born with a brain or head that's too big, and you have microcephaly when the brain and the head is too small. What's going on molecularly? Like what mechanism is messed up that causes that to happen? And so I'm using fruit flies because there are a lot of conserved mechanisms between flies and humans.